Hello everyone, welcome back. Today I'm here with the seventh part of this question series, and in this part I will discuss questions from economic botany and biotechnology. Let's dive in. So the first question for today is: the cosmic vector is made up of. Our options are eastern plasmid, bacteriophage and plasmid, artificial chromosome, or bacteriophage. So its right answer is bacteriophage and plasmid. It is made up of lambda phage and plasmid. As we know that plasmid is the extra nuclear DNA which is present in certain bacteria, and it is combination of this lambda phage and plasmid of bacteria. So the combination of these two things will produce this type of vector which is known as the cosmid. And next, so the vector is a piece of DNA that is transferred to the host cell to replicate desired segment of gene or DNA. Then there are certain features of a vector. Vector के कुछ features होते हैं, like first it should have origin of replication, जिसमें ORI present होना चाहिए. It enables stable maintenance and replication within the host cell, क्योंकि जो desired segment of gene है उसको replicate करना है host cell के अंदर. So this is very important point. That is origin of replication. Then it should have cloning site, that is it should has MCS, that is multiple cloning sites. So there are features: restriction enzyme recognition sites for the insertion of foreign DNA, means ऐसी sites जहाँ पे हम जो desired fragment of gene है उसे वहाँ पे insert करते हैं. The next it should have selectable marker. So ये जो gene है ये help करती है identification of transformed cell or non-transformed cells by antibiotic resistance. For example, we have certain bacteria which have resistance gene for amplicin antibiotic. So ये जो gene है that will act as a selectable marker. The next, it should have reporter gene that facilitates screening of clones through expression of reporter gene. The next, it should have expression elements. It includes promoters, ribosome binding sites for clonal DNA expression. So these are some features that a vector should have. The next, we have different types of vectors. Like we have the most common one is the plasmid vector, and among plasmids we have this one that is called PBR322. This is the most commonly used plasmid as a vector, and this PBR stands for P stands for plasmid, B is for Bolivar and Rodriguez, and 322 is the serial number given to this plasmid. Then this PBR322 was enhanced by adding certain features, and this is named as PUC, that is produced by University of California, and this is the serial number that is given to this. Then we have this cosmid vector that I have already discussed, and phagemid vector again. Then bacteriophage vector, we have artificial chromosomes like BAC and YAC. So these two vectors which are mainly used to replicate large genome sequences. The next, we have list of plasmids, list of vectors and the insert size they can contain. Firstly, we have this lambda phage. Host is ecology and it can replicate up to this much size of DNA, that is 5 to 25 kb. Then we have cosmid. It can replicate insert size of 35 to 45 kb. Then we have this phagemid vector that can replicate a size of 70 to 100 kb of foreign DNA. Then we have BAC. It can again replicate a size of 100 to 300 kb of genetic material. Then we have bacterial artificial chromosomes that can replicate a size of genome or DNA less than 300 kilobases. Then we have this yeast artificial chromosomes, and its host is Saccharomyces cerevisiae, and it can carry a genome sequence of 200 to 2000 kilobases, means it can carry this much foreign DNA and can replicate it. Next question is from PCR. The annealing of DNA primers in PCR requires a temperature of, our options are 50 to 60 degrees Celsius, 94 degrees Celsius, 74 degrees Celsius, or 70 to 80 degrees Celsius. So its right answer is 50 to 60 degrees Celsius. As we know that PCR, it stands for polymerase chain reaction, and this is a rapid method for specific DNA amplification within a DNA template. And this technique was developed by Gary Mullis in 1985. And its basic principle is thermal cycler. It is based upon the principle of thermal cycler. It involves 3 steps. The first step is denaturation, which is done at very high temperature, that is near about this 94 degrees Celsius. It involves separation of the 2 DNA strands. Then second step is the annealing, which is performed at much lower temperature as compared to the denaturating temperature, that is 45 to 60 degrees Celsius. So at this temperature annealing is done, and annealing involved addition of this primer to the DNA, which provide a site for DNA polymerase, or we can say the enzyme used for this PCR tag polymerase, which is obtained from a bacteria, that is Thermus aquaticus. So this is the enzyme which is stable at very high temperature, and this RNA primer provides site of attachment to this enzyme for the next step, that is the third step, that is known as extension, which involves polymerization of DNA. So this step involves 1 cycle of PCR application, and feasibility of PCR depends upon thermostability of polymerase, and these polymerases are obtained from microorganisms, like we are using this TAC polymerase, that is Thermus aquaticus, and it is stable up to a temperature of 94 degrees Celsius. And this extension is done at lower temperature, that is near about 70 to 75 degrees Celsius. So for denaturation it requires high temperature, that 94 degrees Celsius. For annealing it requires temperature of 50 to 60 degrees Celsius, and for extension it requires temperature of 70 to 75 degrees Celsius. And we have different types of PCRs. One is the real-time PCR, and second one is the reverse transcriptase PCR, that is RTQ PCR. So the purpose of annealing in PCR is to allow primers to bind to specific complementary sequences of single-stranded template DNA, and this step is crucial for efficient and specific binding of primers to the target sequences, enabling the subsequent extension of DNA polymerase. Means basically this primer provides site of attachment to this enzyme, that is DNA polymerase, to generate new DNA strands. So after many cycles it can produce huge amount of DNA from a small amount of DNA. So this involves amplification of small amount of DNA or specific DNA into a large amount of DNA. Next question is, azolla is used as biofertilizer as it has. Our options are cyanobacteria, mycorrhiza, rhizobium, or large quantity of humus. So its right answer is cyanobacteria. It contains BGA named as anabina. So this anabina helps in nitrogen fixation. As we know that azolla is the smallest pteridophyte, it is also known as water fern. So the leaves of azolla are inhabited by this blue-green algae, that is anabina or cyanobacteria, and makes it as effective biofertilizer. So the leaves are inhabited by these anabina, that is blue-green algae, and it contains one enzyme that is known as nitrogenase, which makes it a strong, powerful nitrogen fixer and enhance the nitrogen content in the soil, and also improves the growth of plants. So azolla is used as biofertilizer because it contains the cyanobacteria. It contains one enzyme that is known as nitrogenase, which allows them to fix atmospheric nitrogen into the form that is easily absorbed by plants, and it increases availability of nitrogen in the soil, boosting plant growth, and it also improves the organic content in the soil. So that's why these cyanobacteria, that is anabina, is the main reason for azolla being as a powerful fertilizer. Mycorrhiza is association of fungi with the roots of higher plants, and there are several types of mycorrhiza association, like we have arbuscle mycorrhiza here. The fungus produces small arbuscles or vesicles which penetrates into the cortical cells to absorb the nutrients or nutrition from the plants, and this fungus belongs to group Glomeromycota. This is very important. It belongs to this group that is known as Glomeromycota. Then we have another type of mycorrhiza, like we have ericoid mycorrhiza, monotropoid mycorrhiza, and orchid mycorrhiza. In case of orchids, the seeds are very small and contains very less amount of endosperm, so they require accession of mycorrhiza for its germination. There are certain crops and associated inoculant rhizobiums, like we have fava bean and field bean. Here, inoculant used is rhizobium. Then chickpea, we use Mesorhizobium cicer. In soybean, the rhizobium used is Bradyrhizobium japonicum. Lentil, again rhizobium. And alfalfa, it is Ensifermili loti. This is the rhizobium which is used in alfalfa. The next we have common bean. Here again rhizobium is used. Cowpea, Bradyrhizobium. Melcani is used. Then we have groundnut. Here again rhizobium species are used. So these are used as inoculant, which helps in increasing nitrogen fixation in beans, legumes. The next question is SCPs commercially obtained at present from. Our options are Mycona and other bacteria, Moschella and Agaricus, yeast and Fusarium, Chlorella and Agaricus. So its right answer is yeast and Fusarium. In addition to this, certain bacteria, Chlorella, which is a single cell algae, these organisms are also used for production of single cell protein. Then we have this bacterium that is Methylophilus or Methylotrophus bacterium, that is again used for production of single cell protein. So this SCP stands for single cell protein, and it is referred to as refined edible protein. That is one of the alternate sources of proteins for both humans and animal feed. So it is extracted from the pure microbial cultures, and it includes dead or dried cell biomass of microorganisms like bacteria, algae, and fungi. And for this, we use certain substrate which are carbon rich, and the fermenter, we use certain microbes which include fungi, algae, bacteria, and yeast for production of this SCP, which fulfill the protein requirement of humans as well as animals. The next question is, who gave the definition of biotechnology? Our options are EFB, that is the Europe Federation of Biotechnology, National Center for Biotechnology Information, National Institute of Health, and National Center for Cell Sciences. So its right answer is the Europe Federation of Biotechnology. This is the organization who gave the definition of biotechnology. So biotechnology is a science-driven industry sector that uses living organism and their derivative to produce products and processes. So this is a multidisciplinary field integrating natural and engineering sciences. It involves application of organisms, cells, parts thereof, and molecular analogs for products and services. And this term was used in 1919 by Carl Eric to describe the production of products from raw material with the aid of living organisms. And the definition of biotechnology was given by, that is, the Euro Federation of Biotechnology. Next question is, which of the following is appropriate method for cryopreservation? Options are over solid carbon dioxide at minus 79 degrees Celsius, low temperature deep freezers at 80 degrees Celsius, minus 80 degrees Celsius, in vapor phase nitrogen at minus 150 degrees Celsius, or in liquid nitrogen at minus 196 degrees Celsius. So cryopreservation is done with the help of this liquid nitrogen at minus 196 degrees Celsius. So this is the most common method to preserve certain germ plasm for future purposes. So cryopreservation is the process of preserving cells, tissues, and other biological samples by freezing them at very low temperatures, commonly at minus 196 degrees Celsius, using substances like liquid nitrogen. The technique helps to halt all biological activities within the cells, allowing them to survive freezing and thawing processes. Cryoprotective agents like glycerol, salts, and sugars are added to samples to reduce the freezing points of the medium and prevent ice crystal formation, which can damage the cell membranes. So these are the processes which are involved in cryopreservation. We use certain cryoprotective agents like we have glycerol, salts, or sugars. Next question is, International Center for Genetic Engineering and Biotechnology is present in Delhi, Lucknow, Pune, or Chennai. So this institute is located in New Delhi. That is International Center for Genetic Engineering and Biotechnology. and mostly the center is known for research, training, and technology transfer to industries in the field of biotechnology and promotes sustainable global development. The next question is Arteria cardamom belongs to family. Our options are Gingiberaceae, Lauraceae, Myrtaceae, or Iridaceae. So this spice that is on a cardamom, commonly known as queen of spices, it belongs to family Gingiberaceae. And the other spices which belongs to this family are one is ginger and other is curcuma longa. So these spices also belongs to this family. The next this is the photograph of this cardamom, and here the part used is fruit or seeds, and it contains near about 80 essential oils, volatile oils, which makes it very effective spice. The next we have this one spice that is commonly known as ajwain, and its scientific name is Trachispermum emi. His scientific name is Trachispermum emi. It belongs to family Apiaceae. That is, again, it is a member of family coriander family, that is Apiaceae. And here part is used again fruit. The next we have this spice named as Asafoetida, commonly known as hing, and this is again a member of family Apiaceae, and this is the photograph part plant, and its root is used for the production of this resin substance, resin exudate, that forms this spice which is very pungent. It contains certain sulfur compounds, and its name is Asafoetida. Its scientific name is Ferula asafoetida. It belongs to family Umbelliferae or Apiaceae. Again, and the oleoresin is obtained by increasing the lining of the rhizomes at roots. It contains certain chemical substances like we have isobutyl propanyl, that disulfide resin. Then it is mostly used as carminative, expectorant, antispasmodic, or laxative. Expectorant means medicinal substance that helps to overcome the cough conditions. The next spice, that is the king of spices, this is the photograph of Piper nigrum. Piper nigrum, that is Kali Mirch. And here again part uses fruit, berry. It is a member of family Piperaceae. Family is Piperaceae. And here the part used is fruit. And this spice is commonly known as king of spices. It is also known by the name black gold. The next we have this spice that is known as capsicum or chili. Its scientific name is Capsicum annuum. It belongs to family Solanaceae. It is a member of family Solanaceae, and here the part used is fruit. In case of chili, we use the fruit as spice. The next we have this chili contains certain chemical like we have capsaicin and dihydrocapsaicin, and its power is indicated by this scale that is known as the Scoville heat index. The next we have this spice that is known as cardamom, large cardamom, and its scientific name is Amomum subulatum. Its scientific name is Amomum subulatum, and again member of family Gingiberaceae. It belongs to family Gingiberaceae, and here the part used is fruit seed. Again the part used is fruit seed. In Hindi we call it as badi ilaichi. The next we have another spice named as cinnamon, and its scientific name is Cinnamomum zelanicum, and it is a member of family Lauraceae. It belongs to family Lauraceae. Here the part used is bark. We have another plant part belongs to this family, and the name of genus is again same, that is Cinnamomum tamala, and this is Tejpatta. So it belongs to same genus. Family is again Lauraceae, so this is again a spice, and this is cinnamon. The next we have this important spice named as Schizium aromaticum. In Hindi we call it as long, or in English we call it as a clove. Here The part used as spice is the floral buds, which are harvested and dried and is used as a spice. So this is clove, and it belongs to family Myrtaceae. It's a member of family Myrtaceae. Here the part used is floral bud. Use the floral bud as spice. The next we have cumin seeds. In Hindi we call it as jeera. Again, it's a member of family Apiaceae and part used is fruit. Next we have fennel. In Hindi we call it as saunf, and its scientific name is Foeniculum vulgare. Again, it's again a member of family Apiaceae, which is a coriander family. It's a member of family Apiaceae. This is known as fennel. Here again, part used is fruit. The next we have this spice, which is very costly, named as saffron, and its scientific name is Crocus sativus. It belongs to family Iridaceae. It's a member of family Iridaceae. And here the part used are the female part of the flower, that is style or stigma. The next question is experiment to prove the synthesis of organic compounds formed on the basis of origin of life. This is a question from evolution. Our options are Haldane, Operin, Miller, or Fox. So this first evidence of synthesis of organic compound was given by the experiment which was performed by Miller and Urey. So this is the right answer. So Miller and Urey experiment, they showed that in primitive earth conditions there were these gases, when water, the methane, ammonia, or hydrogen. From these gases, under this primitive earth condition, there was formation of organic compounds. So in this Miller and Urey experiment, Miller and Urey used an experimental setup in 1953 to recreate what environmental conditions were believed to be likely on the Earth: a gaseous chamber simulator, an atmosphere with reducing compounds such as we have methane, ammonia, hydrogen, an electrical spark with high voltage near about 800 degrees Celsius temperature. This simple apparatus caused chemical reaction that produced a variety of organic molecules, and some of which are basic building blocks of life, such as amino acids. And although the exact atmospheric condition of early Earth are still debated, the Miller and Urey experiment provided the first evidence that organic compounds or molecules could be spontaneously produced from inorganic matter. So this was the first experimental setup which showed the synthesis of organic compounds from inorganic substances under primitive Earth's condition. The next question is genetic drift is a change of. Our options are gene frequencies in the same generation, gene frequencies from one generation to next generation, appearance of recessive gene, or none of them. So its right answer is gene frequency from one generation to next. So this genetic drift is a fundamental principle in evolutionary biology. This is a part of evolutionary biology. It is a stochastic, non-selective process of frequencies